time, the incidence of drug-resistant infections, in particular multi-drug resistant infections, is rapidly increasing, to the point in some clinical settings we're seeing 60% of infections are multi-drug resistant. So antibiotic resistance is a thing, it's bad, and we should do something about it, is basically the crux of the issue. A common way that bacteria become resistant to antibiotics is, obviously we treat with antibiotics, we pretty much do exactly what you would do to select for resistance in a laboratory setting, and lo and behold we get resistance. And one of the ways they do that is by overexpressing efflux pumps. So you get proliferation in the face of antibiotic treatment with antibiotics, and treating with more antibiotics doesn't resolve the infection. So efflux pumps are these sort of 3 protein systems that bifurcate the cell wall of the bacteria, and they basically are there to throw things that the bacteria doesn't want inside of its cytoplasm back out into the environment. So your antibiotic goes into the cell, and the efflux pump throws it back out. You throw more antibiotics at the bacteria, and you select for increased expression of those efflux pumps, and the bacteria becomes resistant to your antibiotic of choice. Fortunately, we, and by we I mean Chen, discovered a lytic phage in a pond in Connecticut called Dodge Pond that infects Pseudomonas aeruginosa. I'm the bioinformatics nerd on the project, so it has a 242 kb genome, so it's relatively big for a phage. It's in the Syphoviridae family, and it's a 5kz-like virus. So it's kind of one of these big, fat-headed standard lunar lander module bacteriophages. So what OCMO1 is what we named the phage, or I think Paul named the phage that, does is it forces P. aeruginosa into an evolutionary trade-off. So we treat the bacteria with phage, they lyse a bunch of those phage, however resistant strains emerge, and those proliferate in the face of phage. What's really cool about this is that Ben did some really elegant knockout experiments with knockout strains of aeruginosa, found the binding site for this phage is actually the outer membrane protein of the efflux pump. So what the phage does is binds to efflux pumps and forces selection in the opposite direction to antibiotics, in that the resistant strains that emerge after phage infection don't express efflux. What this potentially allows us to do is treat with a combination of phage and antibiotics to push the bacterium into an evolutionary corner in which we can kill multi-resistant Pseudomonas, which I think is a really cool idea. So does it work? It's obviously the $50 million question here, at least in the lab. So this data is really cool. I really like this graph. At least in the lab, we think it does. So the black line here is growth of Pseudomonas with the presence of, as a control. In the gray line that actually goes above our control is the growth of Pseudomonas in the presence of the antibiotic tetracycline. So it's basically laughing at the antibiotic and growing better than it does in the control. In the blue line is the growth of bacteria in the presence of our OCMO phage. And as you can see, it largely suppresses it until the end of the experiment when you start to see the emergence of these viral resistant strains. And when you combine tetracycline and the phage, you actually don't have growth above the level of detection. So I think this really convincingly shows that a combinational therapy, at least in the laboratory in a theoretical setting, works well. What was super cool is that it works across a variety of antibiotics that are efflux. So this is a series of antibiotics, and this is the improvement in fold of that antibiotic in the presence of phage. And what's really cool is in the presence of ciprofloxacin or Cipro, which is one of our last resort antibiotics, we're seeing a 20 times improvement of that antibiotic in the presence of virus. And tetracycline, which is obviously why we showed the nice graph, is 60 times better in the presence of phage. So at least in the lab, we should know that this is a really promising virus for treating Pseudomonas infections. And really, you come to, that obviously brings to the question of, does this work in a clinical setting? And fortunately, through the FDA compassionate care approval, we actually had a patient who was willing to let Ben and I inject them with viruses. Whether or not that's a smart idea, I don't know. So this patient had an aortic arch graft. So this piece of the aorta had been replaced with a DACON implant. And following surgery, they developed a Pseudomonas aeruginosa infection at the base of the aorta, which is a place you really don't want a Pseudomonas infection. Suppressive doses and high doses of IV antibiotics had not had any effect on this infection. It had spread to the sternum, so he had to have part of his sternum debrided, so removed. He had a fistula form from his chest cavity out to the surface and was leaking delicious Pseudomonas juice down his shirt. And he'd approached multiple cardiologists who had refused to operate based on the risk of spreading that infection. So he really wasn't in a good state. So surgical procedures were out of the question and antibiotics weren't working. We treated this patient with a bacteriophage cocktail in January. So this was OCMO1, the phage we found that causes this evolutionary trade-off, along with a highly virulent strain of phage to knock them down, and a high dose of antibiotics to finish the job. Now, Ben, who was the driving force behind this treatment phase of the project, and our cardiologist, Deepak, have a great story about trying to administer the phage to the actual side of the basal side of the infection with a needle through the chest cavity. However, there's too much scar tissue following the debridement of the sternum, so they have a great Pulp Fiction-esque story about trying to jam a needle through some guy's chest. As an alternative, we administered the phage through the fistula that had formed to the outside of the chest. So, following up with this patient, did our treatment work? Patient left the country soon after treatment. Fortunately, we'd spoken to the patient's family, and they had surgery to remove that fistula in April. And that means two things that we think are quite promising. The first is that we didn't kill him. So I was a bioinformaticist on this project, and I was handed an Illumina and PacBio dataset and said, make sure there are no toxin genes in this dataset because we're going to inject this organism into a patient's chest. And that adds an extra level of stress to your standard bioinformatic analysis. Also, Ben needed to purify this phage in order to satisfy the FDA guidelines for administering it through compassionate care, and we managed to do that to a point where we didn't kill the patient. Yay. Also, the fistula removal surgery would not have been possible if that fistula was still draining tons and tons of Pseudomonas goo. A surgical procedure to remove this fistula would not have been possible if there was still major infection present. So this concludes us to optimistically say that this maybe worked. And I'm sorry that I don't have a more conclusive finding at the end of my talk for you guys. For future directions in this, I have a graduate student who's giving a talk later in the session to using a bioinformatic approach to determine how likely broad spectrum is this approach, how conserved are efflux pumps, and how likely is it that we can treat different Pseudomonas infections using this particular phage approach. Can we evolutionary walk this phage using experimental evolution approaches on mixed host environments to work in other bacterial efflux pump systems? Can bacteria escape this trade-off and potentially render it useless in the future? I don't know, is the honest answer to that question. And can we find phage that force other trade-offs for other resistant mechanisms? And where do we look for those phages? Is an open question. I just started my lab last summer. Last summer at UC Merced, and we had a bunch of other projects because I'm mostly a bioinformatics nerd. So SMART's major project is looking at the microbiome and symbiosis of cone snail venom ducts. We also have some JGI-funded work looking at mimulus microbiomes, looking at how the microbiome changes and influences plant responses to drought, landscape genetics and antibiotic-resistant bacteria in the Central Valley, mostly sequencing sewage from wastewater treatment plants. The field works awesome. Doing some gain of resistance experiments in bacteria that we acquire through feedlots and other high-intensity agriculture throughout the Central Valley, and looking at gain of resistance experimentation. And we're also looking at lizard skin to see whether it has any antimicrobial properties in multi-drug resistant bacteria. And if I haven't bored you to tears and you're looking for a graduate spot, come talk to me. Thank you very much for listening, guys. Yeah.
So, one patient?
One patient.
Any follow-up, like any intentions to follow up and try others? Or?
Yes. So
the question is, we only have one patient. Are we intending to treat others? Yes. So the mechanism we've been using to try and circumvent the whole clinical trial stage, which is obviously prohibitive in terms of time, money, expense, is the compassionate care track to treat patients. And we do have a second patient that we're potentially talking to about using a phage-based treatment. So hopefully, and we're talking to, one of the other challenges with phage therapy is that you can't patent a phage. So in convincing the pharma companies to fund or become interested in phage therapy work is difficult because they can't patent the treatments. So I think it's going to have to be a grassroots approach to pushing this up. Yep.
So in the lab, you went to 16 hours. Is anybody working on going further than that to see if resistance develops with combined therapy?
Yes. So Caitlin, who's the rotation student, is pushing this out to 2 weeks. What she's mostly looking at is whether or not the suppression of efflux expression continues after you remove the virus. And that does appear to be the case. So it potentially opens the window up between using phage and using antibiotic. As to whether or not they escape the combinatorial therapy, I think is an open question. And I honestly don't know the answer there. They probably can, because they do stuff we didn't think of. Jason?
Yeah, you tried anything with just like the contractile kill of the actual functional phage? Like, is it possible to, if you had that mechanism, to the efflux pump and somehow block the effectiveness of it?
Yes. So the question is, can we just use the phage tail rather than the actual virus itself as a mechanism to block efflux and force this trade-off to occur? And the answer is yes, that's something that we're thinking about and we are intending to explore in the future. Well, mostly John Wirtz's lab is intending to explore in the future. So yes, it's a good point and it's something we have